Okay, so here are the answers for the Unit 2 Triple Biology Higher Tier 2018 paper. The first question says, the photograph below shows a young ice skater, Tracy, who falls and fractures her leg. The diagram below shows 2 ways in which Tracy's fractured leg could be treated in a large modern hospital. Give one reason why Tracy might choose traditional surgery and one reason why alternatively she might prefer to be treated by an injection of stem cells. So with traditional, she's able to stand sooner, because if we look here, traditional, it says it's possible to stand after 2 days, whereas with injecting, it's 3 weeks. And then stem cells then, however, with stem cells then we have a faster healing percentage over time, 82% compared to the 59%. When fractured bones heal, cells divide and multiply by mitosis. Complete the following table. Chromosomes in mother cells 46, they've told you the number of daughter cells in mitosis is 2, and the number of chromosomes is 46 because it's the same, they are genetically identical to the mother cell. Following mitosis, what must happen to stem cells in order for them to repair the fractured bone? They must differentiate or specialise. Say the name of the disease that can occur if cell division by mitosis is uncontrolled. It can cause cancer, and stem cells can be obtained from both adults and embryos. Give one reason why some people have a strong personal objection to the use of embryonic stem cells, and the only thing you need to write down is it destroys potential life. The photographs below shows the appearance of an eye before and after a reflex action, which occurs in response to a change in light intensity. Label A and B on the diagram. So A is the pupil and B is the iris, the muscle in the eye. From the photographs, describe how and why parts A and B of the eye alter when the light intensity changes. So in brighter light, the iris contracts because it's a muscle, so it can contract to reduce the pupil size, which we can see. This then restricts the light entering, so the retina is protected. So 2 features of all reflex actions, they are protective and they are automatic. Josie investigated reaction time in humans. She tested 3 people, 2 males and 1 female, ages 10, 30 and 50 years old. They had between 1 and 20 years experience of playing the keyboard. By means of computer app, 90 random music notes flashed one by one onto a screen. The person being tested then instantly pressed each note on the keyboard as soon as it was seen. Each person did the test 3 times. No incorrect notes were pressed. A data log recorded the number of notes which were pressed within 0.2 seconds of appearing on the screen. The bar chart shows the results of the investigation. Use the bar chart to answer the following questions. How does repeating the test affect the reaction time? Will we get faster reactions or the reaction time it decreases? Calculate the percentage change between test 1 and test 2 for age 50. Give your answer to 1 decimal place. So at age 50, test 1 we had was 10, and then test 2, 15. So 10 divided by 15 times 100 is 66.7%. From the data, what 2 conclusions could you make about the effects of age on reaction time? That the older the person, the slower the reaction time. After practice, older people improve more than the younger people. Josie decided to try the investigation again, and to make a fairer test of the effects of age, state 2 variables which you could control. So we're looking at gender, the handedness, if they're left or right-handed, and their eyesight. So one other way in which the investigation could be improved is to get a larger sample size. Describe the structure and function of DNA. So DNA is 2 strands. Those strands are made up of an alternating sugar and phosphate, which are connected by bases. And the 4 bases are adenine, thymine, cytosine and guanine, and they form a double helix. The base pairing of adenine, thymine, cytosine, guanine is that A binds with T and C binds with G. We have the triplet code, where 3 bases code for a specific amino acid, which makes proteins. Basiliximab is a monoclonal antibody used to suppress the immune system of kidney transplant patients. It works by preventing white blood cells from attacking the cells of donor organs and therefore reduces the probability of rejection. Apart from rejection, state another disadvantage of kidney transplants is that there's a shortage of donors. Explain why and how white blood cells of the recipient would attack the cells of the organ donor, therefore leading to rejection. Because On the donor kidneys, there's going to be antigens. The lymphocytes are going to produce antibodies, and they're going to attach to the foreign antigen and attack it. The diagram below shows the process of producing monoclonal antibodies. So we've got the mouse injected with the antigen. We've got cells B and cells A, cell C, the cultured medium that only one cell allows cell C to survive, and then produce monoclonal antibodies. Use a diagram and your own knowledge to answer the following questions. State the name of the cells labelled. So A are our cancer cells, B are our lymphocytes, and C are our hybridomas. State how the following cells were produced. So B are produced by the injected antigen, and it causes that immune response of the lymphocyte production. The hybridoma then, lymphocytes and cancer cells are fused together, which forms our hybridoma. State 2 other examples of medical uses of monoclonal antibodies for diagnosis of disease and for pregnancy testing. Students investigated factors affecting leaf growth of ivy. They measured the leaf stem length and the leaf width of 50 leaves of an ivy plant growing on the south side of a tree trunk at a height between 1 and 2 metres. They compared these measurements with ivy leaves of the same ivy plant growing on the north side of the same tree trunk at the same height, and also leaves from the same ivy plant growing flat on the ground. North side hasn't got a lot. The results are shown below. Calculate the ratio of the leaf stem length to leaf width for the ivy growing on the north side of the tree. So we've got our 60 divided by our 52, okay, which gives me a ratio of 1.15 to 1. Suggest why there's a difference between the ratio of leaf stem length to width length for the ivy growing on the ground and for the south side. It's because it's got more light, more access to water, and a higher temperature. Students recorded the data for the ratio in the frequency table below. State the type of variation shown by the ratio. It would be continuous variation. The diagram below represents the human nephron. The concentration of various substances is shown at 2 different points, A and B. Use a diagram and your own knowledge to answer the following questions. Name and describe the process occurring at point C. So point C is here. I just got the diagram before. So it's ultrafiltration. And the small molecules move from the capillary into the Bowman's capsule. Explain why the concentration of glucose is different at points A and points B. So point A and then point B is because all of the glucose has been selectively reabsorbed into the blood capillaries. D, state the name, is our proximal convoluted tube, so our PCT. State a reason for the increase in the concentration of urea and mineral salts between A and B, and that the water then has been reabsorbed at that point. Some students investigated the tropic responses of the dandelion. What is meant by a tropic response? It's the growth of the plant in response to a stimulus. The students set up the apparatus shown below on a south-facing windowsill. The students concluded that the dandelion had shown positive phototropism. Suggest why the students cannot be confident in their conclusion. Explain how they could change the method to improve the confidence in their conclusion. Well, they haven't used AA control. The plant is subjected to both light and gravity. Set up the same experiment of the dandelion inside the dark to test one variable to improve the confidence in their conclusion. State the name of the plant hormone responsible for phototropism, and it is auxin. Penicillin is an antibiotic that is produced by several species of fungus from the genus Penicillium. Its effect was first identified by Alexander Fleming in 1928. It was only in 1939 that penicillin was extracted by Howard Florey and Ernest Chain at Oxford University and later developed for use as an antibiotic. The industrial scale production of penicillin was developed in the USA between 1941 and 1943 in response to the high number of soldiers who were dying due to diseases linked to their wounds during World War II. Penicillium can be grown industrially in a tank called a fermenter. An example is shown in the diagram opposite, which is here. State how penicillin helped save the lives of wounded soldiers. Well, it killed the bacteria and prevented them from getting infections in their wounds. Name a group of microorganisms that would not be affected by antibiotics. It would be viruses, because antibiotics only kill bacteria. State a danger of new drug treatments, that there are possible unknown side effects. We don't know what effect they're going to have. Use a diagram of the fermenter and your own knowledge to answer the following question. Other than nutrients, state one factor that affects the growth of penicillium and state how it can be controlled. So pH, and that's why we have our reservoir to control how acidic or alkaline the liquid is, the solution, and temperature. So you've got a water jacket to maintain the temperature of the fermenter. The graph below shows the quantity of penicillin and penicillium inside the fermenter at 25 degrees over a period of time. Using the graph, calculate the mean growth rate of penicillium between 20 and 50 hours. So penicillium, so we went up, and then we had 15 divided by 30, which gives me an answer of 0.5. Identify the optimum time for the penicillium to be extracted from the fermenter and to justify your answers. Any time between 96 and 100 hours, because the mass of penicillium is at its maximum. Draw a line on the graph to estimate the effect of increase in temperature from 25 degrees to 28 on the dry mass of penicillium produced between 0 and 120 hours. So you'll see that it will increase quicker, but it will reach the same end point eventually. Dylan set up an experiment to study negative feedback mechanisms associated with rapid cooling of the hand. A temperature sensor was held between the fingertips of a volunteer and another sensor was taped to the skin on the upper chest. The sensors were connected to a data logger that was set to record for 3 minutes. The volunteer placed their right hand into ice cold water 30 seconds after the recording started. The hand was then removed from the ice cold water after a further 30 seconds. The results from the data logger are shown below. State what is meant by negative feedback mechanism. So it's a change from the normal conditions resulting in the body restoring normal conditions. Describe the results for sensor A after the hand was placed in the ice cold water, and you can see that the temperature decreases. Explain the change in temperature recorded by sensor A between 30 and 90 seconds, that's the decrease. So receptors detect the drop in temperature, so the stimulus. The blood vessels vasoconstrict, or they get narrower, so less blood flows to the skin, so less heat is lost. Suggest the reason for the increase in body temperature recorded in sensor B between 30 and 90 seconds, and it's because more blood remains in the core of the body and not on the surface of the skin. Suggest how the results of the experiment would differ if the volunteer had consumed an alcoholic drink 20 minutes before the start of the experiment. The response to change in temperature would be slower. Varroa destructor is a parasitic mite of honeybees. The colony of honeybees affected by the parasite becomes weakened and eventually dies out. The parasitic mite originated in USA, where it is the pest of the Asian honeybee. It is spread across most continents, arriving in the UK in the early 1990s and affecting the native European honeybee. Use your knowledge of classification to suggest whether parasitic mite was able to spread from Asia to the Europe honeybee. And the reason it is, if you look, that the European has a genus of Apis, and so has the Asian honeybee. So they have the same genus, which means they're closely related. So the mite would enjoy both of them. In 2014-2015, there were 1,650 registered beekeepers in Wales who kept bee colonies, each of which is one beehive. The National Bee Unit recommends that honeybees be treated with pesticides to try to reduce the number of parasitic mite in beehives. In recent years, many beekeepers in North West Wales have stopped treating their bees with pesticides. These beekeepers believe that there's no difference in colony losses between the bees treated with pesticides and bees not treated with the pesticide. The table below shows the winter losses between 2010 and 2015 in North West Wales. What percentage of beekeepers in Wales took part in the survey? And it was 5%. Use the information in the table to state one conclusion about the effect of the pesticides of winter bee losses, is that using pesticides actually causes more winter losses. Give 2 reasons why some beekeepers across the UK would argue that the strength of evidence is weak. And it's weak because only 5% of beekeepers, there's not a lot to compare. It was only carried out in North Wales, nowhere else in the UK, so it doesn't represent other parts of the country. Early attempts to control the parasitic mite involved using chemical. By the late 1990s, the parasitic mice had developed resistance to the chemical, and the beekeepers were advised to use other chemicals to control the pest. Explain why over time the chemical became less effective at killing the parasitic mite. Well, the mite had mutated, which gave rise to the resistance in the pesticide. It allows the ones with the resistance to survive and the ones without to die. This is known as survival of the fittest. The ones that survived were able to reproduce and pass on the gene to the next generation, which is then natural selection.